All right, it is 2, so let's get started. So for test two grades should be up, so you should be able to see that. You can't see your test right now, so I'll make that available probably by tomorrow. What I'm going to do is I'm going to like just go over some of the more difficult problems that seem to give people a lot of trouble. I'm going to make a YouTube video about that, release that video, and then open up test 2 so you can look at that at the same time, see how you did, see what you got wrong. Yeah. So for the test, if you remember way back at the beginning of the semester, I did say that I will drop your lowest test. So what's going to happen is once I release that test 2 video, I'm going to more or less just drop your lowest test right now so you can see an accurate representation of your grades. And as we go on to tests 3 and 4, I will continue to update what is the lowest test so you can always have a up-to-date view of what your grade in the class is. So that pretty much covers everything I need to say on test 2 as of right now. So yeah, let's get into the actual material. So remember, this is all for test 3. And if you remember way back on Wednesday, we've been looking at example proteins and we looked at muscle contractions. We're looking at the power stroke here. We're also looking at myosin and actin, the 2 major proteins responsible for the power stroke. And we also talked about the regulation by triple myosin and troponin. And so we had this question about rigor mortis, how you need ATP for the power stroke. And just one more question that is a good refresher from what we went on Wednesday. And this is a question I often see people misunderstanding. So let's take a look at it. So what type of interactions are involved in the binding of myosin head to actin? Are there any covalent bonds made or broke in the cycle of reactions that move the thick filament relative to the thin filament? So the big confusion that I generally see for this problem is what is actually a covalent bond? So if you remember way back to general chemistry and organic chemistry, right, that's a covalent bond when elements are actually bonded. However, for myosin to actin, they're all non-covalent bonds. During the power stroke mechanism, the way myosin and actin interact, they don't form any bonds with each other. It's all hydrogen bonds, polar interactions that are holding these 2 molecules together. So the type of interactions for myosin to actin are all non-covalent. But there are covalent bonds being broken during the cycle of the power stroke. If you remember way back to Wednesday when we were talking about the power stroke, the main energy from the power stroke is coming from ATP. So ATP is being bound to our myosin and it's hydrolyzing to ADP plus phosphate. So I'm just going to draw out ATP really quick here. So instead of drawing out adenosine, I'm just going to say A is adenosine. There's AMP. There's ADP. So this is the molecular structure of ATP. And every time you remove an inorganic phosphate, which is that PI just means a phosphate unit, you're breaking off one of these. So when we are turning ATP to ADP, we always break a covalent bond to release one of these inorganic phosphates. right? So that's the answer to question 6 there. So any questions about that, or anything anyone had about the power stroke? I know that was a while ago, back on Wednesday, but anything at all? Right. So we have one more thing to talk about myosin until we move on to antibodies, which I have scheduled for today. Or sorry, not myosin, actin. So we said that actin is part of muscle contraction, but actin has a big role in the protein, or sorry, in the cell outside of muscle contractions. And remember, we have 2 types of actin. We have G-actin, which is globular. That's being shown here. And we have F-actin, which is fibrous, which is being shown here. And so what can happen to actin is that they can form this fibrous actin, and they can do a process called treadmilling, where they can make this actin fiber move by removing actin from one end and adding it onto another end. And this is how the cell can like expand or reach, as being shown here. So this cell is trying to get these nutrients, right? And so it is morphing itself to reach over and get them. That is 100% due to actin that's helping to reshape the cell. So the actin is the cytoskeleton for the cell, but it is constantly reforming. It's not like our skeleton, which is just stuck in place. The cytoskeleton can reform, and it does that by actin. Now, fibrous actin has 2 ends, right? Because it's a line. You have a plus end and the minus end. And G-actin can actually bind to both, as being shown in this picture. So we have G-actin coming on here. We have G-actin coming on here. And they can come off from both sides. So you see we have a G-actin coming off from the plus end here, and a G-actin coming off from the minus end. However, there is certain affinities, right? G-actin really wants to bind the plus end, hence the plus end, and it wants to come off the minus end. And we can see that in this picture, right? So on the plus end, we have 6 actin going on, while on the minus end, we only have 1 going on. At the plus end, we have 1 G-actin coming off, while at the minus end, we have 4 coming off. So in general, this thing will treadmill in this direction. So actin will start to come off on the minus end and will appear on the plus end, and that's how the cytoskeleton can be mobile, right? So what actually happens going from this globular actin to this F-actin during this treadmill process? Well, what happens is that just globular actin, G-actin, has ATP bound to it. And when it becomes F-actin, that ATP is hydrolyzed to ADP plus phosphate. And in its ADP form, this actin really does not want to be around other actin. And so it actually takes a little bit of time. So let me erase some of this to make this easier to see. So this picture is really nice to follow along. So you can see that here we have like this blue actin adding on to this plus end, but after a while it will end up in the middle because of that treadmilling. And it takes a while for that ATP to hydrolyze. So as soon as you're added, you don't necessarily hydrolyze right away. You'll take a little bit of while to hydrolyze, and then you want to be removed. Well, if you keep adding to one side, you're more likely to remove on the other side, the minus end. So that's why actin generally come off the minus end, because at this point, they're mostly already hydrolyzed. You mostly have ADP actin. While here, you can still have ATP actin, and this ATP actin still will want to interact with other actin molecules. Like I said, you can have this treadmilling or a steady state where the addition and removal of actin is at the same rate. So this fiber actually never changes in length, it just changes in position. And that's what that treadmilling is, what I've been mentioning this whole time. It's like the belt on the treadmill, where the belt never actually changes length, it just changes position. That's how an actin fiber works. And as shown in this picture, and as I already mentioned, it can stretch the plasma membrane, allowing for extension of cytoplasm in any direction. And that's one way that a cell can move. So that is one of the other important uses of the actin protein. So any questions about anything on this slide or anything at all? All right, if not, I'm going to switch over PowerPoints to the one I prepared for today, as this should be that is the last slide for the PowerPoint we had on Wednesday. So let me just quickly switch here. The PowerPoint I had prepared for today, where we're going to continue to talk about examples of protein function. And we're going to move to the immune system and how the proteins in the immune system help to keep you happy, or not happy, healthy. So we're going to go over a very rough view of the immune system. Those of you who've taken immunology, you probably know a lot more about this stuff than I do, but we're going to be focusing on the actual proteins here in a minute, but let's just go in the rough overview. So how does your immune system become active? It responds to a molecule called an antigen. And an antigen can really be anything. It's just something that an antibody reacts to. For us, an antigen will usually be a foreign macromolecule, either a protein or a sugar. It's going to be your most common antigen. Now, the way that this works is that you have beta cells, and on the surface of your beta cells, you have immunoglobulins or antibodies. And let's say this beta cell attaches to an antigen. What will happen then is that it will eat this antigen antibody complex, and then a T cell will help to stimulate that beta cell, and you'll start making antibodies. And you will also create memory beta cells. So if the same antigen is present or presented to you, months or years later in your life, you will already have these memory beta cells ready to go and ready to help protect you against the same type of infection, which is kind of being shown here in this plot. So the y-axis of this plot is days. And here in the red, we have our first encounter with antigen A. And you can see after 7 days, we have, like 100 antibodies ready to go. And we'll keep that up. And then the infection dies down, so we make less antibodies. But let's say we see that same antigen a second time a month after we first saw it. Since we have those memory beta cells, we can skyrocket our antibody production. They're already ready to go to help us really fight against the second coming of antigen A. While if a new antigen, antigen B, is presented to us, it will have the same curve as antigen A. Since this is the first time we ever saw it, it takes us a little while to get our antibody production up the speed. While antigen A, this is the second time we saw it, so we can really speed up the production of these antibodies. So that is our very, very, very broad view of how the immune system works. So let's actually get into some of the specific proteins that make the immune system happen. And we're going to be focusing on the human immunoglobulin G. There's a lot of immunoglobulins. I mentioned those in the videos you were supposed to watch for today. We're mainly going to focus on G though. And G is our Y-shaped friend here. So when we look at immunoglobulin, there are two different fragments. The FAB fragment, which makes up what I like to imagine as the crab claws, so here and here, and the FC region. So the FAB regions, the Fab regions, they're identical to each other. So you have two of those, and you have one FC region. Now, if we look at the FAB region, you have what's known as the light chain and the N-terminal half of the heavy chain. So light heavy. So the light chain is up here, and the heavy chain is this thing. So you can see why they got their names light and heavy, hopefully. The light chain is a lot lighter in weight than the heavy chain. So you have two light chains and two heavy chains. It's on this FAB region that we're going to try and bind the antigen right here in our antigen binding site, right at the end of our little crab claws. That's where we're going to bind the foreign macromolecule. And the FC region has the C terminal of our heavy chains. FAB region has the N terminus. FC, by the way, just means crystallized. That's because a lot of these names were discovered before we actually knew what this looked like. So in the different experiments people ran, they found out, oh, one chain is lighter than the other one, light heavy chain. And one section of the region of the antibody crystallizes a lot easier than the other one. And so what researchers did is that they added this molecule papin. What papin does is that it will cut our antibody right here. And this is proteolysis. Let's break down this word proteo. Basically means protein. Lysis means to cut. So proteolysis is simply a reaction that cuts protein. So they add this molecule to antibodies, and they find, okay, one region crystallizes super easily. So that's the FC, and the other one doesn't. That's the FAB, right? That's where these names come from. That place where it's being cut is called the hinge region. That's connecting the FAB and the FC. And along with, light chains, heavy chains, FC, FAB, one other type of naming scheme we have are the variable regions and the constant regions. And that's color coded for us. And that's what the V stand for. So the pink part of our antibody are variable regions. That means their amino acids change all the time, right? They can have like any amino acids in here, while the constant region, which is yellow, doesn't change. They remain constant. And one beta cell will only produce one type of antibody. And there are millions, tens of millions of different antibodies that your body could produce. So you need to have a B cell for every type of antibody you could produce. So any questions about the information presented here on the antibody? So just so we understand how this like protein is made and connected back to how you might see this protein using different experimental approaches, I want you to tell me, what are the approximate molar masses of our immunoglobulin if we analyze this protein using gel filtration, using SDS page and draw a gel and label it, and if we use SDS page in the presence of 2-mercaptan ethanol. So this is a little bit of review of our protein techniques. So I'm gonna give people a couple minutes and try to solve this for me. Some information you need to know is that light chains are gonna be 23 kilodaltons and heavy chains are gonna be 65 kilodaltons in weight. So see if you can figure out A, B, and C, connecting information that we learned about like almost a month ago now to our antibody structure here. If you have questions, let me know. Otherwise, I'll be back in a couple minutes to help walk you through this if you get stuck. All right, so hopefully you got at least somewhere with that. So let's remember what our techniques are. So gel filtration is just a technique used to weigh a protein, and it doesn't degrade the protein. So the antibody should come out as one complete unit. So the weight from our gel filtration would be as 2 light chains. So 2 times 23, so that's the weight of 2 of our light chains, plus the weight of our heavy chains, 2 times 65, because we have 2 heavy chains. So the total weight of our antibody is 176 kilodaltons. So that's the way we would see our antibody through a gel filtration column. And SDS page, so this is for B and this is for C. If you remember SDS page, small things are at the bottom, big things are at the top. And if we don't add 2-mercaptoethanol, all SDS page does is denature this molecule, but denaturing does not mean breaking covalent bonds. So the antibody would stay as one protein. And so we'll get, oh, that's really ugly. We'll get one band at 176 kilodaltons. So just like gel filtration. However, if we add 2-mercaptoethanol, what 2-mercaptoethanol does is that it breaks disulfide bonds. So every disulfide bond we have will be broken. And the only place this matters is connections, right? So we have a disulfide bond that's connecting the light chain and heavy chain. That's broken. So the light chains are now free. So we'll get one bond, or one band rather, at 23 kilodaltons for our light chains. And the disulfide holding the heavy chains together is broken. So again, we'll get one band at 65. So if you do an experiment with immunoglobulin G with and without 2-mercaptoethanol, this should roughly be the results you would expect to find. One band at 176, and then in the beta-mercaptoethanol, one band at 23, one band at 65 kilodaltons. So any questions about this idea here, or our review on techniques in biochemistry? All right. So let's actually look at the secondary structure of our immunoglobulins now. And they have what's called the immunoglobulin fold, where you have 3 to 4 antiparallel beta sheets that form a sandwich that are linked through a disulfide bond. So the way to look at this picture, this orange, that's the disulfide bond. And these blue and red, these are your beta sheets, and they're forming the sandwich that and you can bind your antigen between our variable regions here. Now, where we really see the variation is our hypervariable loops. So these loops out here, these are the things that are changing. And they're right at the tips of our FAB region. If we go back to our picture here, it's where all this squiggly stuff is, right? So that's what they're trying to show with these squiggly lines. Those are the hypervariable loops. And these are what is changing from antibody to bind different antigens, right? So what this means is that when we bind an antigen, it is completely non-covalent. The only thing that is dictating how we bind is hydrophobic interactions, hydrogen bond interactions, and ionic interactions. And if you remember again, ionic interactions are like salt bridges, positive and negative charges interacting. And antibodies are extremely specific in that they will only bind one thing, but they'll bind that one thing really strong. KDs 10 to the minus 4 times 10 to the minus 10. Yeah, I'm sure, like, since you've never looked at KDs, those numbers probably don't mean a lot to you. But if you get like lower than 10 to the minus 6, that's like super strong binding. So a number like 10 to the minus 10 is just astronomically strong. It's like Gorilla Glue times a million strong of binding. It's going to grab it and it's going to have a hard time letting that antigen go. Now, if we look at IgG sense, instead of going back to it, I'm just going to redraw it. Looks like our Y, right? There's my crappy IgG. It's divalent because it has two variable regions, so it can bind two things at once. You have other antibodies that I talked about in the videos, like IgM and IgA are multivalent. They can bind multiple things at once, but IgG is only binding two things at once. And antigens, it's possible for antigens to have multi-binding sites for different antibodies, right? Because if you imagine a giant protein, like an antibody could bind here, and a different antibody could bind here, and a third antibody can bind here. Remember, an antigen is simply something that an antibody will bind to. You know, it could be a protein, it could be a carbohydrate sequence. So a specific macromolecule could have multiple antigen binding sites. And what that means is that your antibodies, when they come in contact with antigens, they often form a lattice, a cross-link lattice. And that's what's being shown here, right? So these pink regions are our antigens, and the symbols are actually what the antibodies are binding to. So I guess to be more specific, like the triangle, the circles, and the squares are our antigens. These colored Ys are our antibodies. And you can see we're binding two things at a time, and we have multiple antigen binding sites. So we're linking together all of our foreign macromolecules into this one big meshwork. And this is going to make it easier to clean up and destroy these antigens if they're just floating along all together, outside of the cells. So that's the strategy that's going on here. And being divalent really makes this happen, where you can bind two things at once. And if you bind different parts of the protein, you just make this huge meshwork lattice. Right, so any questions about the information presented on this slide? All right. All right, so our second question here. Why is variation only in the variable loops of our IgGs? In other words, why don't we see variation in the FC region, right? And I'm not looking for it because the hypervariable loops are in the variable region. I'm trying to think of big picture evolutionary scale. Why would it make sense that you only have variation in one point and not the other point? So take a minute, think about that, see if you can come up with some logic to that, and I'll be back in a minute. All right. Let's go back and look at this logic then. So the FAB region, this is going to be binding to our antigen. So it makes sense why that should be variable. You never know what antigen you're going to come up against. So you want to be ready for everything. But the FC region, this is going to be interacting with beta cells, right? Your own body's cells. Your own body cells, they're not going to change. And you want any antibody to interact with that specific beta cell. So since this is interacting with your body, your cells, it makes sense why we want to keep that constant so it can be recognized. While this is interacting with antigens, and you never know what antigen you're gonna face. So that's why that is always kept variable, and the FC region is always kept constant. It really just goes back to thinking about, okay, what do we want to interact with? So next question to more or less continue our understanding here of why antibodies work. So why do some antibodies raised against the native protein fail to bind to the corresponding denatured protein? And this goes back to really our whole discussion on how proteins fold. So imagine that you have an antibody, and that antibody only binds to an alpha helix. It's looking for that alpha helix. Well, in the denatured, so this is native, in the denatured version, that alpha helix would become an IDP. And so you no longer have the helix to bind to, so you'll no longer bind to that protein. A more extreme example is, let's say, like, this is really weird because I'm not really good at drawing, but let's say your folded protein looks like this, where let's say this is residue like 12, and this is residue, I don't know, 123. Oops, let's do that better. That's residue 123. And so when you're looking at the primary sequence, residue 12 and residue 123 are separated by 113 amino acids. However, when that protein folds, these two amino acids are now very close in space. And let's say your antibody binds to that region right here. So it binds to where antibody 12 and 123 meets. However, when the protein would become denatured, 12 is right here, 123 is over here, you don't have the same local conformation of amino acids anymore. Your antibody can't bind. So that's why, you can bind to a folded protein, but if you show an antibody that same denatured protein, it's not going to work. So any questions about that idea? All right. And let's see, do I want to do that one? We can skip question 4. It's just getting at how these frameworks form, but we can just skip that. I think this question is really good to check your understanding again, but just for time's sake, so we can make sure we don't fall too far behind during the week and the rest of the semester. We can just skip question 4. So the last big topic, the last topic I want to talk about for today is autoimmune diseases. Now, antibodies, the way that antibodies should work is that they'll bind to foreign molecules only. And that's pretty amazing when you think that your body has tens of thousands of different molecules in it, yet you will not produce antibodies that will attach to these molecules. Well, normally you won't. If you do, you'll start to get a disease, as shown down here that are the different diseases due to your antibodies attacking your own tissues. And the mechanism for eliminating antibodies that attack your own tissues is not well understood. But it has to do with eliminating those beta cells and T cells that would recognize your own body, right? So during development, somehow, some way, we eliminate any beta cell that would attach to your own material. And this happens from the moment you're born onwards, even moment when you're first developing your immune system. But like I said, if anywhere along the way you lose this ability to get rid of beta cells that attack your own cells, that's when you get these autoimmune diseases. And when you look at it, some tissues are attacked more than others. And it's possible that you can get an autoimmune disease through some kind of injury or some kind of disease there. Probably one of the most common types is rheumatoid arthritis, which is your body attacking your own connective tissues, causing inflammation and degeneration of joints. One that sounds really bad is lupus, because there you are showing you have produced antibodies towards DNA or phospholipids, and DNA and phospholipids are in every one of your cells. So you're going to start in attacking every one of your cells. You're going to start saying that these are foreign molecules that need to be destroyed, which is not a good place to be. And that gets us to our last question about the systematic lupus erythematitis, where you produce antibodies to DNA and phospholipids. So normally, like I said, why would you not make antibodies to this? Because every cell has DNA, every cell has lipids, and you don't want to say to your immune system, every cell in your body is foreign and needs to be destroyed, right? Now, so that's A, that I kind of explained just a minute ago. And B, so during a normal response to a viral or bacterial infection, the immune system produces a large amount of antigen-specific antibodies, and the resulting complexes are removed from circulation and degraded. Here, for SLE, we see a large antigen-antibody complex accumulate in tissues that aren't cleaned away. Why is that? Again, it's going back to this idea that DNA, phospholipids are in every cell. Your antibodies will just be going crazy at that point, attaching on to everything, all your phospholipids, all your DNA. You're not going to have time to clean these out because there's just so much of it, and you'll just have buildup of antigen antibody complexes and that whole mesh we were talking about in the previous slides. You're just going to have that in your own tissues, and it's really going to be a big problem. But yeah, that ends our PowerPoint for today on our journey into looking into antibodies. I will say antibodies are used a lot in experimentation when you're trying to see if you have a specific protein or macromolecule present. You're going to use antibodies to make that determination. So is there any questions whatsoever about anything we went over today? All right. If not, that's all I have. So I'll put a homework up like always. Thank you for stopping by or watching YouTube. I appreciate it. And hopefully I'll see people on Wednesday. Have a good day.